Hello, my name is Tom. I'm a postdoc in the Cancer Genomics group led by Peter Van Loo at the Francis Crick Institute in London. Our lab develops new method to better understand how tumors initiate and evolve through somatic alterations, selective pressure, evolutionary sweeps, and tumor heterogeneity. In this context, copy number alterations, aka CNA, are a key process in which the genome is subjected to gains and losses. As shown in this figure, the transformation of a normal cell into a cancer cell often leads to drastic changes in the genome, and many chromosomal aberrations can be seen here. For instance, this cancer cell only has one single copy of chromosome 2, whereas it has 4 copies of chromosome 4. At a larger scale, almost all cancers show evidence of CNAs, although chromosomal instability broadly varies across cancer types. This figure here shows the global level of CNAs in 33 cancer types on the x-axis, and we can see some cancer types that are prone to extreme degrees of instability, such as sarcomas and ovarian cancers, compared to low instability in leukemia. To study the genomic landscape of CNAs, our lab developed ASCAT, which stands for Allele-Specific Copy Number Analysis of Tumors. ASCAT accurately assesses CNAs from cancer genomes, where the major and the minor alleles are respectively shown in red and blue. In the first profile, the cancer genome is flat and all chromosomes have 2 copies, whereas in the second profile, we can see a very high level of chromosomal instability with a lot of gains and losses. Originally, ASCAT has been developed for SNP arrays, but we have aimed to extend its scope, especially for sequencing data, whole genome sequencing, whole exome sequencing, and targeted sequencing. And over the years, we further pushed ASCAT to a cutting-edge tool to call CNAs. Its segmentation has been improved, including a multi-sample processing. Log-OR can now be corrected for GC content and replication timing. There is an improved calling method for chromosome X, etc., etc. Now, to show that ASCAT is a platform-independent CNA caller, we considered cases included in the TCGA and PEACOCK studies. In TCGA, nearly 12,000 cases had SNP6 and whole exome data. We also generated pseudo-targeted sequencing data by downsampling whole exome data to 568 genes of interest. And in PEACOCK, more than 2,000 cases have whole genome sequencing data, and a quarter of them are also part of TCGA. Although PEACOCK provides 50 targeted sequencing, including 40 TCGA cases. We first compared resolution across platforms, and this figure nicely illustrates that the resolution broadly varies across the platforms. As expected, whole genome sequencing has a high resolution with almost 1 million heterozygous SNPs, whereas pseudo-targeted sequencing data has the limited resolution with a bit less than 1,000 heterozygous SNPs. In this figure, we are comparing purity ploidy fits, and the top right corner is about purity and the bottom left corner is about ploidy. Overall, we observe very good agreement with a median absolute purity difference of 1.5 to 5% and a median absolute ploidy difference of 0.01 to 0.14. We then compared copy number alteration profile from ASCAT using 2 metrics of interest. One is the allele-specific Manhattan distance expressed in millions of base pairs, and the other is the intersample agreement expressed as the fraction of the genome with the exact same copy number status. So in this example with 2 profiles from different platforms, the Manhattan distance is 114, which is a bit less than half of the chromosome 1, so it's quite good, and the intersample agreement is 97.20%, which is quite good as well. This figure summarizes Manhattan distance and intersample agreement values for all combinations of platforms, and the median values are shown in the table on the right. We can see that most platforms perform very well, and the intersample agreement of more than 95%, except the pseudo-targeted sequencing, where 90% agreement are measured, likely because of a low sequencing coverage that is about 60x. Ultimately, this allows us to study the landscape of CNAs across cancer types, and this figure summarizes key metrics of interest, including purity, ploidy, fraction of the genome with the loss of heterozygosity, fraction of cases with a whole genome doubling, and the number of segments. We can go even further and use CNA profile to characterize tumor evolution and heterogeneity in great detail. And we know that the transformation of a normal cell into a cancer cell is a multi-step process with key genomic alterations, allowing cells to proliferate. By leveraging copy number information, we can characterize what and when such alterations occur, as well as quantify tumor heterogeneity through cancer subclones. And I would like to describe 3 such downstream analyses here. The first analysis is about building phylogenetic trees from CNAs. And in this example, I'm using MEDIC2 on 2 samples, one primary tumor and a matched metastasis. MEDIC2 builds phylogenies based on the most parsimonious route that explains gains and losses between samples. And here it helps identify shared clonal alterations versus the one that are sample specific. The second analysis is about reconstructing the subclonal compositions of cancer. Here I use dpCLAS to correct variant allele frequency for copy number and purity, allowing me to get cancer cell fraction estimate. Somatic mutation or cluster based on the CCF values informing about the proportion of cancer cells carrying such mutation clusters. Ultimately, this allows building subclone trees across 3 tumor samples, 1 primary tumor and 2 matched relapses. The third analysis is about timing copy number gain. So if a mutation is located in a region where there would be a gain, then its multiplicity increases. However, if the mutation happens after the gain, then it's simply not copied. So this principle allows pinpointing when copy number gain occur, including whole genome doubling during tumor's evolutionary past. In this example, we observe a gain on chromosome 1 and 5, and using GRITIC, we can infer when such alteration occurred. Chromosome 5 is an early event, then a whole genome doubling occurred, and chromosome 1 is a late event. So all combined, ASCAT accurately assesses somatic and allele-specific copy number changes in cancer genomes across platforms, and CNA is a central entry point into covering key aspects of tumor initiation and evolution. As for the perspectives, other datasets are currently being analyzed to further benchmark our methods. We will also release all ASCAT profile on TCGA cases for the scientific community, and some extra feature will soon be added, like processing sequencing data for unmatched tumor and or non-human sequencing data, etc. Now, thank you for listening to my short talk, and I'm looking forward to the conference. Bye!